Hello students. So today we are going to start with a new topic, that is biotechnology. Now in this topic we are going to learn basics of biotechnology, the traditional biotechnology as well as the modern biotechnology, the principles and process of biotechnology, the technique of gene cloning and rDNA technology, PCR, that is polymerase chain reaction, the biological tools which are required. Also, we may learn further the cloning vector, competent host, and methods for rDNA technology. Okay, now in the present slide, as always, keep the animation with relation to the topic. You can see how gene therapy can take place, the DNA strands which are joined by gene manipulation or genetic engineering, etc. Also below you can see how with the help of robotics we can do a lot of work related to research, and also the biotechnology can be used for healthcare sector as well. Okay, now coming to the next slide, the introduction part of biotechnology. It is a product of interaction between the biological science and technology. From the name, or we can say definition, the biotechnology itself means biological science and technology. That is two different parts merging together. That is biology and technology for the benefit of mankind. It is in fact an applied branch of biology. So now biology is also a part, or can say branch of biology, and also further we come across more branches in future, like genetic engineering or gene methodology, or few other which are on the process, bioinformatics, etc. The term biotechnology was used by Carl Ericke in the year 1990 to describe a process for large-scale production of pigs. So it was initially used by Carl Ericke. Further, biotechnology. It is broadly defined as the development and utilization of biological forms, products or processes for obtaining maximum benefit to man and other forms of life. As I mentioned, biotechnology helps to develop and also utilize all the different biological forms, products, process, etc., which actually benefits the mankind and also other forms of life. According to OECD, that is Organization for Economic Cooperation and Development, in the year 1981, it is the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and service to the human welfare. It uses scientific principle of microbiology, genetics, biochemistry, chemical engineering, mathematics, statistics, computers, and industrial processes. So in biotechnology we also use all these principles which helps to make the process much more easier. Biological agent means plants and animal cells, microorganisms, enzymes, or their products. So when you say biological agent, it is none other than the biological forms, whether it can be in the form of a plant cell, animal cell, or microorganisms, including their enzymes and the products as well. So history of origin of biotechnology is as old as human civilization. Development of biotechnology in terms of its growth occurred in two phases, that is traditional and modern biotechnology. That is, this biotechnology we are using a terminology in the last 10 to 15 years, but actually biotechnology is a age old process, right from our grandparents or great grandparents, or when the civilization started, this biotechnology process was started or was going on. Only thing is we did not know what form of technology is being used, what a scientific process is going on. Now as we come to know about the detail, so we can put it as a traditional biotechnology, as we can say homemade methods or methodology, and the modern technology using the techniques or modern forms, or using all the principles of engineering, etc. Okay, here again I put an image where you can see how the genes by CRISPR work, how the joining of the genes or changing of a gene could take place by genetic engineering or gene therapy. Now, as we learned about traditional and modern, so we come across what is traditional, what happened in the age old process, what our grandparents used to do or great grandparents used to do. That is, it was primarily based on fermentation technology using microorganism, as in the process of preparation of curd, ghee, soma, vinegar, yogurt, cheese, winemaking, etc. So as we know, at home we prepare curd, we prepare the vinegar or yogurt or cheese, etc. at home itself by simple methodology, simple process. Now, during 1970, a new technique of recombinant DNA technology was developed and then established by Stanley Cohen and Herbert Boyer in 1973. So these two scientists established a new technology in the field of DNA, this is recombinant DNA technology. The technique permits to change or modify genetic material for getting new specific products. Just in the previous slide, as I showed, the CRISPR method, where you can modify or change the gene by genetic engineering or gene therapy. The combination of biology and production technology based on genetic engineering evolved in modern biotechnology. There are two major features of technology that differentiate modern biotechnology from the traditional one. So how the modern and the traditional are differentiated, or there's a difference between both, we can put up in two simple points. Capability of science to change the genetic material for getting new specific products through rDNA, that is recombinant DNA technology. Polymerase chain reaction is PCR, which we are going to learn in the further slides. Microarrays, cell culture and fusion, and bioprocessing. Secondly, ownership of technology and its socio-political impact. Now, as in the modern technology, the products which are made, the ownership, or we can say biopatent, etc. is also being carried out, which we'll be going to learn in the later part of the chapter. Now, the convenient or conventional industries, pharmaceutical industries, agro industries, food industry, etc. also focusing attention to produce biotechnological based products. So you can see here in traditional biotechnology how bread making, etc. was carried out. Further, now we learn the principles and process of biotechnology from the basic difference between traditional and the modern part. So what are the principles? Modern biotechnology is based on two core techniques, which genetic engineering and chemical engineering. So these are two important aspects or two important core techniques used in the principles and process of biotechnology. Now, genetic engineering deals with alteration of genetic material, that is DNA and RNA, while chemical engineering deals with maintaining sterile environment for manufacturing variety of useful products including vaccines, antibodies, enzymes, organic acids, vitamins and therapeutics. Now vaccines, you know, present situation, vaccine is an important part to overcome all the viral infection, or in case of the COVID, right? Antibodies, again to overcome any type of viral fever or bacterial infection. Enzymes, organic acids, vitamins, therapeutics, etc. The genetic engineering is defined as the manipulation of genetic material towards a desired end and in a directed and predetermined way used in vitro process. So here genetic engineering helps to manipulate with the help of the genetic material which is present and what we desire or what we require accordingly by a in vitro process. That is, we can say in simple words, the in the lab condition, in the uh, test tube using pipettes, beakers, etc. in the lab condition which we can prepare. Manipulation of genes involve repairing of the defective genes or replacing of defective genes by healthy genes or normal genes. Artificial synthesizing of a totally new gene transfer of gene into a new location or into a new organism, introducing an altogether new gene manipulation of genes for improvement of living organism, combining of genes from two organisms, altering the genotype or gene cloning, etc. So why this manipulation is carried out? One, to remove the defective ones. If defective ones are done, then it will remain for lifelong. So because of genetic engineering, the defective gene can be replaced by adding up the normal gene. Also, we can transfer the gene from its particular location. We can create a new organism. You can manipulate the gene for improvement of the living organism, or you can combine two genes again from two different organisms by altering the genotype, or we can do gene cloning as well. Therefore, the genetic engineering is alternatively called recombinant DNA technology or gene cloning. So hence the name rDNA, recombinant DNA, where the genes are manipulated, or the genes are replaced, or transfer of totally new genes in a new location. So again, just as I mentioned, you can see in one of the images where by in vitro process how it can be carried out in lab condition, or in the second image you can see how with the recombinant DNA technology we can use it for various purpose, whether it is for DNA cloning or gene transfer, or again in case of any cell RBCs, or in the lab condition for preparing of antibiotics, vaccine, etc. Now, little bit of additional information. Now, first you can see the image, how the DNA structure is there, or first you can identify the particular DNA or DNA strand which you want to change, which you want to transfer, the location, etc. Then second image you can see how in a human body the genome can be transferred or alteration can be carried out, right? So here John E. Smith in 1996 gave definition of genetic engineering: the formation of new combination of heritable material by insertion of nucleic acid and molecule produced by whatever means outside the cells into any virus, bacterial plasmid, or other vector system, so as to allow the incorporation into a host organism in which they do not occur naturally, but in which they are capable of continued propagation. So it was John E. Smith who defined the term genetic engineering. Okay. Then further, the technique of gene cloning and rDNA technology, how it can be done. Now again, before going to the points, just see the image. As I always tell in biology, if you see the image or diagram, the learning becomes easier, understanding becomes more easier. So if you see in the image towards the right part, recombinant DNA technology, first the DNA is isolated from two sources and cut by the same enzyme, that is plasmid vector and gene of interest. Then some of the uses of genes, how it is inserted into a recombinant DNA, and then it can be inserted, or you can see used in a transgenic organism for gene for toxic cleaning bacteria, gene for pest resistance, or protein to stonewash the gene, or protein for dissolving the clots, or some uses in proteins. Also towards the left side also you can see how the DNA cloning is carried out. You can remove the particular gene from the plant, and then the DNA structure can be cloned, or it can be inserted into a plasmid and a recombinant is formed, or we can insert into a plasmid. Now again, what is this plasmid? Plasmid is usually present in a bacterial cell, which helps to replicate a DNA. Okay. Now, coming to the points. In gene cloning, a gene of known function can be transferred from its normal location into a cell via suitable vector. Now, as I told, the vector, which is nothing but the plasmid, which has a capacity to self-replicate, which is autonomous, that is functions on its own, or it can duplicate itself. The transfer gene is replicated normally and is handed over to the next progeny. Okay. Now, coming to the tools and techniques for gene cloning, rDNA technology. Now when you say tools, now for anything and everything, when you make certain things, even for construction, you need various tools. So even in case of gene cloning or rDNA technology, we need tools. So again, coming to the image which I put up, where you can understand what are the basic tools required. First, the vectors, agarose gel, restriction enzyme, or we can say cut DNA, modified enzymes, remove or add the particular gene or phosphate or nucleotide, ligase to join, or we can say simple words, that is the gum which is required. In case of restriction enzyme, the scissors, polymerase synthesize DNA and RNA and add or remove nucleotides, synthetic DNA, oligonucleotides, synthetic genes, polymerase chain reaction, that is PCR. Now coming to the points, the basic requirement for techniques are different instruments or device. Macromolecules such as DNA, RNA, proteins, etc. are synthesized in the living cells, which vary in their molecular weight, solubility, presence of charges, absorbance of light, etc. Several techniques are used to isolate and characterize the macromolecules. The size of the different types of molecules varies, and therefore their molecular weight also varies. The technique used on basis of molecular weight are gel permeation, osmotic pressure, ion exchange chromatography, spectrometry, electrophoresis. Now these are the different techniques used in the rDNA technology, and these are different methods, we can say, as a tool: chromatography or electrophoresis or spectrometry. Further, electrophoresis, what is that? It's a separation of charged molecules applying on an electric field. It is applied for separation of DNA, RNA and proteins. Now with this you could understand very easily electrophoresis, that is, it helps to separate the required DNA, RNA or the proteins. Further, DNA being negatively charged migrates to anode. Small fragments of DNA molecules move faster and thus separate faster. Use of agarose gel electrophoresis, PAGE, SDA, PAGE are the different methods of electrophoresis. Now when you say what is electrophoresis, separation of charged molecules, but even in that there are different methods like agarose gel electrophoresis, PAGE or SDA PAGE. Now coming to next part, polymerase chain reaction or PCR. Now you can see the machinery which is used for PCR, that is polymerase chain reaction. It is another device used for gene cloning or gene multiplication. In vitro, it is amplification of gene of interest through PCR. So what is done here? Simple words, that is, you make more copies of or gene cloning, or you multiply the gene by the in vitro method. Now in 1985, Carey Mullis made an important discovery in the form of extremely powerful technique, that is polymerase chain reaction, which is PCR. Now PCR can generate a billion copies of the desired segment of DNA or RNA with high accuracy and specificity in a matter of few hours. That means by using this particular method, we can make billion of copies in just few hours. Okay. The process of PCR is completely automated and involves automatic thermal cycles for denaturization and renaturization of double-stranded DNA. The device required for PCR is thermal cycler. So you require thermal cycler, that is a device which is used for PCR. PCR is in vitro amplification of desired DNA segment, which requires DNA containing the desired segment to be amplified. Several molecules of 4-deoxyribonucleoside triphosphate, that is DNTPs, excess of 2 primer molecule, heat stable DNA, polymerase and appropriate quantities of Mg plus ions, that is magnesium ions, are also required. Now coming to the mechanism of PCR, how the process takes place step by step. At the start of PCR, the DNA segment and excess of 2 primer molecules for 4-deoxyribonucleoside triphosphate and the thermostable DNA polymerase are mixed together in Eppendorf tube and the following operations are performed sequentially. Step 1. The reaction mixture is heated to a temperature 90 to 98 degrees centigrade to separate two strands of desired DNA. This is called denaturation. What is the first step then? Denaturation. How it is carried out? By heating it at a temperature. What is a temperature? 90 to 98 degree centigrade. Why it is used? To require or remove or separate the desired DNA strand. Clear? Now what is the second step? Annealing. What is that? The mixture is allowed to cool 40 to 60 degrees centigrade. That permits pairing of the primer to complementary sequence in DNA. This step is called annealing. So what happens over here? After the first step of denaturation, here in the second step, annealing, the mixture is first allowed to cool again at temperature of 40 to 60 degree centigrade, which helps in pairing of the primer or the complementary sequence in DNA. Third step is what? The prime extension. That is, the temperature 70 to 75 degrees centigrade allows thermostable, that is Taq DNA polymerase, to use single stranded DNA as a template. Template means, we can say in simple words, copies, and adds nucleotides. This is called prime extension. It takes around 2 minutes of duration. Further, one cycle takes around 3 to 4 minutes to begin. Second cycle, DNA is again heated to convert double stranded DNA into single stranded DNA. Further, in an automatic thermal cycler, the above three steps are automatically repeated 20 to 30 times. Thus, at the end of n cycles, 2n copies of DNA segments are produced. The machine performs the entire operation automatically and precisely. Once the desired number of cycles is completed, the amplified DNA segment is purified by gel electrophoresis. After its sequencing, the amplified DNA can be inserted into a cloning vector. Desired gene can also be obtained from the gene library. What is gene library? Where the required gene is stored. So whenever you require, you can easily obtain it. So what are the three steps? First is denaturation, second annealing, and third prime extension. What is denaturation? It is heated to a temperature of 90 to 98 degree to separate the two strands of DNA or desired DNA. Second is annealing, where it is allowed to cool for 40 to 60 degrees centigrade, where the primer pairing up of the complementary sequence of DNA takes place. And thirdly, the temperature 70 to 75 degrees allows thermostable Taq DNA polymerase to use single stranded DNA as a template or copy and adds the nucleotides. Okay. Now, just an image where you can see the copy of desired DNA. The first step, as we just saw in the previous slide, denaturation 90 to 98 degrees centigrade, DNA strand separated, that is in the second stage. Annealing of primer 40 to 60 degrees centigrade, replicating DNA strands using Taq polymerase 70 to 75 degrees centigrade. Third, copies of desired DNA, that is renaturization. Second cycle continues to form 4 copies of desired DNA. Cycle continues to produce 2 copies of DNA. So that is how the DNA replication through polymerase chain reaction or PCR technique is carried out. Now, biological tools. What are the tools required for it? Now, just as I told in the beginning, or we saw the slide, the tools or accessories like scissors, gum, all these are required. So here also we have biological scissors, biological gum for carrying out all the process. So what are those? There are 3 types of biological tools used: enzymes, cloning vector, that is vehicle DNA, and competent host for transforming with recombinant DNA. As just we learned the vehicle DNA like plasmids. First one, enzymes. Different enzymes include lysozymes, nuclease such as exonuclease, endonuclease, restriction endonuclease, DNA ligase, DNA polymerase, alkaline phosphate, reverse transcriptase are used. Now, first one, restriction enzymes. Now, enzymes that cut the phosphodiester bonds of polynucleotide chains are called nuclease. They are of 2 types, exo and endonuclease. As I told in simple words, remember, scissors. So these restriction enzymes are the scissors which help to cut the required chain or the DNA. Exonuclease cut nucleotides from the ends of DNA strands, whereas endonuclease cut DNA from within. So during the 1970s, it was found that bacteria contain nucleases that would recognize short nucleotide sequence with duplex DNA and cut. So this nuclease helps in recognition of the particular nucleotide sequence with the duplex DNA and help in cutting. The phosphodiester backbone at highly specific sites on both strands of duplex is cut by these enzymes called restriction endonucleases or simply restriction enzymes. As I told, in other words, the biological scissors. They were given this name because they are used by the bacteria to destroy various viral DNAs that might enter the cell, thereby restricting the potential growth of the virus. Thus, restriction enzymes serve as a defense mechanism. The bacteria protect its own DNA from the nucleolytic attack by methylating the bases at susceptible sites, a chemical modification that blocks the action of the enzymes. Thus, the restriction enzymes are the molecular scissors that are used to recognize and cut DNA at specific sequence. The sites required by them are called recognition sequence or recognition sites. As I told, simple way to remember the restriction enzyme, that is the molecular scissors which identifies or recognizes and help the DNA to cut at the particular site or the sequence, which is termed as the recognition site or recognition sequence. Further, the different restriction enzymes found in different organisms recognize different nucleotide sequence and therefore they cut DNA at different sites. Okay. Now, again I just put a table which shows a list of restriction endonuclease, the site at which they cleave. So the source and recognition sequence, which is indicated by the arrow of various restriction enzymes. So just in short, I just read out the restriction enzymes first. That is ALU1, BAM, H1, ECO, R1, HIND, 2. So all these are nothing but the bacteria which are used as the restriction enzymes. That is, the ALU is what? Arthobacter luteus. BM is what? Bacillus amelolecufaciens. ECO Escherichia coli RY13, whereas HIND 2 is Haemophilia influenza RD. Okay, then you can see the recognition sequence, the product and the end products. Okay. Now, recognition sequence. Now in the previous two slides we come across the term recognition. What is that? Again, first see the image. Modification or mode of action of ECO R1 to cleave. What is ECO R1? Escherichia coli R1. So that is DNA showing palindromic sequence. Now what does this palindrome means? The mirror image, correct? So you can see the palindrome sequence, then the restriction enzyme ECO R1, and then followed by the action which helps to recognize the sequence. So we go point wise. The sequence recognized by restriction enzyme are 4 to 8 nucleotides long and characterized by particular type of internal symmetry. Consider the particular sequence recognized by the enzyme. So it recognizes the character of the 4 to 8 nucleotides and accordingly it helps in cutting or recognize at which place to cut, or as the term utilized, internal symmetry. Okay, so you can see 3' CTTAAG 5' or 5' GAATTC 3'. When one reads the sequence in opposite direction, 3' to 5' or 5' to 3', it is identical or same. A sequence with its type of symmetry is called palindrome. Okay, I will just give one example which usually we come across, that is the language Malayalam. When you read from front and back, it has the same alphabets, same way in the mirror image as well, correct? Okay, further, when the enzyme ECO R1 attacks this palindrome, it breaks each strand at the same site in the sequence, which is indicated by the arrow between the A and G residues. As you can see, 3' CTT AA, we can see the arrow G 5' and 5' G where the arrow AATTC3'. So restriction enzymes either cut straight across the DNA in the region of palindrome to give blunt ends, or cut producing short single-stranded projection at each end of the DNA to produce cohesive or sticky ends of staggering ends. Further, again, just for additional information here, towards the right you can see Nobel Prize winners in physiology or medicine. There is Werner Erboff, Daniel Nathans, Hamilton Smith, and the types of restriction enzymes: type 1, type 2, type 3, that is the cleavage site, location of methylases, and examples of each. Okay, so if you just read the information, Smith, Nathan and Arbour achieved the discovery of restriction enzymes. For the particular achievement, they were awarded Nobel Prize for Physiology and Medicine in the year 1978. Since restriction enzymes have been used for genetic manipulation by dissecting, analyzing and reconfirming the genetic information at biological or molecular level. Okay, and again you can see the different type 1, type 2, type 3, etc. For example, type 1 which functions simultaneous endonuclease and methylase, which is ECO-K, or type 2 which has separate activities for cleaving and methylation. They are more stable and are used in rDNA technology. Example, ECO-R1, BG2, these enzymes cut DNA at specific sites within the palindrome. Or type 3 which cut DNA or specific non-palindrome sequence, example HPA1, MBO2. There are thousands of type 2 RE enzymes are recognized and discovered. Okay. Now coming to the next part, that is cloning vector or the vehicle DNA. So again you can see here two images I have put of plasmid, which is called as PBR32, whereas bacteriophage vector. So here vectors are DNA molecules that carry a foreign DNA segment and replicate the inside the host cell, that is they make copies inside the host cell itself. Further, vectors may be plasmid, bacteriophage, that is M13 lambda virus, which you can see in the image, cosmid, phagemids, BAC, that is bacterial artificial chromosome, YAC, yeast artificial chromosome, transposons, baculovirus or baculovirus, and mammalian artificial chromosome, MACs. So most commonly used vector are plasmids. So commonly preferred vectors are plasmid such as PBR322, PUC, TI plasmid, and bacteriophage, that is lambda phage or M13 phage. Plasmids and bacteriophage are most commonly used vectors. So these are the one which is commonly used. That's why I put the image on the right hand side, so it will help you to understand what is a plasmid as such and bacteriophage. Further, a good vector should have the ability of independent replication, so that as the vector replicates through ORI gene, that is the origin part, and large number of copies of DNA insert will be formed. Moreover, vector should be able to easily introduce into the host cell. A vector should have marker genes for antibiotic resistance, must contain unique cleavage site in one of the marker genes for restriction enzyme. It should have at least suitable control elements like promoter, operator, ribosomal binding site, etc. The plasmids obtained naturally do not possess all the characteristics. Hence, they are constructed by inserting gene for antibiotic resistance. Example, as you already learned, PBR322, PBR320, PACYC177 are constructed plasmids. PBR32 is most commonly used in rDNA technology in the plant. So out of all these plasmids, the PBR322 is the most commonly used one in the technique. Further, now what is this plasmid? The plasmid are most commonly used in recombinant DNA technology are those that replicate in E. coli. Investigators have engineered these plasmid to optimize their use as vectors in DNA cloning. So you can see again the two images to the left, the plasmid cloning vector showing replication origin, a drug resistant gene, and a region in which foreign DNA can be inserted, which you can see from the image. And also we can see polylinker or HIND3, SPH1, PHT1, SAL, 1, XBL, BAMH1, SMA1, KPN1, SAC, ECO, R1, etc. Similarly, on the right side, simple image showing the parts: promoter, multiplying cloning site, promoter tag, poly A signal, the selectable marker. Now further, plasmid vectors for plants. An important vector for carrying new DNA into many types of plants is a plasmid that is found in Agrobacterium tumefaciens. This bacterium lives in the soil and causes a plant disease called crown gall, which is characterized by the presence of overgrowth or tumors in the plant. The tumefaciens contain a plasmid carrying T1 for tumor inducing. The T1 plasmid contains a transposon called tDNA, which inserts copies of itself into the chromosome of infected plant cell. The transposon with the new DNA can still be inserted into the host cell's chromosome. A plant cell containing this DNA can be grown in culture or induced to form a new transgenic plant. Again, see the image onto the left side, the T1 plasmid, the left border auxin, cytokinin, opine, right border opine catabolism, origin of replication, that is ORI, and virulence region. And you can see on the right side the plant where the T1 plasmid, tDNA, bacterial genome, which is again transformed into the plant cell and which can be used. So it can be used for various purpose according to the need. Then lastly, the competent host or the cloning organism. Now they are usually the bacteria like Bacillus, Haemophilus, or Helicobacter pylori and E. coli. Most E. coli is used for the transformation within the recombinant DNA. So all these E. coli are used for transformation in the recombinant. Also I put the images on the front. You can see the left first, which is rotating image of Haemophilus influenza, in the center E. coli, and towards the right the Helicobacter pylori. So all the three examples of bacteria which are most commonly used in cloning organism. So by seeing the image, we can understand how the bacteria are as such. Okay, so that is the first part, and here are the assignment questions based on today's topic which you have learned. Thank you all of you.